Hello, my name is Eric Brenner, and I'd like to talk to you today about a very important experimental component of the PTC lab, the phenylthiocarbazide lab, where you examine your genotype and see how it is expressing a specific phenotype, the ability to taste or sense this PTC flavor. And that experimental component is called PCR, for polymerase chain reaction. And you've probably heard about it before, and it's something that you will be doing in this lab. So let's make sure that we understand the fundamentals behind this. So first of all, for PCR to work, for the reaction to actually take place, you need very specific components in your test tube. Okay, so let's start out and look at what some of these components are. You need template DNA. Okay, in this case, the template DNA is going to be yours. One part of this experiment is you'll be isolating your own DNA, and you'll be doing the reaction on that to look at your specific molecular phenotype for the PTC gene. So for PCR to work, you also need primers. These are short strands of oligonucleotides, or pieces of DNA that recognize the specific sequence. In this case, it'll be flanking your gene on both sides, and I'll show you a diagram to explain how that works. These primers bind to each side, and they hybridize specifically to given sequences that allow you to PCR that gene. Okay? You need buffer, including various salts, but specifically you must have magnesium, because that operates as a very important cofactor for this, for all polymerases, really. And then, of course, you need the polymerase itself. And for PCR, we use a polymerase called Taq polymerase because, as mentioned in the manual, it was isolated from a thermostable bacteria. And we'll talk about why that thermostability is important too in the next slide. But before we get there, I just want to point out that this reaction can only work if you have a device called the thermocycler, which can rapidly change the temperature in ways that allows the PCR reaction to go forward. So what are those changes that need to occur? So for PCR to work, the temperature needs to go to a very high temperature for DNA denaturation to occur, because you need to break these hydrogen bonds. Okay? You need to separate a strand of DNA into two strands of DNA. Why? Because the next step after denaturation, which occurs at a much cooler temperature, and that makes sense because these oligonucleotides that you've added need to anneal or hybridize. Okay? And that occurs typically between 50 and 60 degrees. And then the next step is primer extension. And so then the PCR machine will warm it up to about 72 degrees so extension occur. And that's the actual polymeration reaction where DNA is synthesized 5' to 3', and you produce your actual new strand of DNA. This reaction is repeated over and over and over again. So with each generation, you double the amount of DNA. And so with doubling, you have logarithmic growth of the amount of DNA that's available. So this is just to illustrate how this works. Again, the TAS2R38 locus is just the nomenclature used to describe the PTC gene. And if you look here, you see that the oligomers bind. They fill in, okay, through the elongation reaction. So after annealing, they elongate to make a brand new strand. Okay, and then that strand here, okay, is melted off again to make new strands. And continuously with additional primers binding, you can then reach saturating quantities of DNA. And that is how PCR works. And I wish you good luck with your experiment.